What is an enzyme? Enzymes are biological catalysts that work to lower the activation energy for a reaction to take place. So how does an enzyme work exactly? For example, when food is eaten, mechanical digestion begins in the mouth, where food is chewed and swallowed, proceeding down the digestive tract. As food enters the small intestine, it is saturated with enzymes. Here we can see the digestion of starch, which is the substrate the enzyme, known as amylase, works upon. An enzyme has a specific shape called an active site, and this is where the substrate binds to. Induced fit now occurs. The enzyme adjusts its shape to better fit the substrate and puts stress on the substrate, causing the bonds inside to break. The enzyme then releases the substrate as two new products, called maltose. It is not always released as two products, there could be any number. In this next example, we see how an enzyme can alter a substrate, and also what parts make up an enzyme. If you consume alcohol, your stomach will get to work digesting the ethanol using the enzyme alcohol dehydrogenase. Enzymes are made of two parts, the large protein structure known as the apoenzyme, and the smaller variable molecules that form part of the active site, called cofactors. Organic cofactors are either coenzymes that are used up in the reaction or prosthetic groups that are tightly bound to the enzyme. Inorganic cofactors can be things such as metal ion activators or iron-sulfur clusters. In this case, our enzyme has both NAD+, a coenzyme, and zinc, a metal ion activator. Cofactors take part in the catalyzed reaction. As ethanol is converted into acetaldehyde, NAD+ is used, releasing NADH. Competitive inhibition can be seen between ethylene glycol, the main component of antifreeze, and ethanol, as both are catalyzed by the same enzyme. Since ethanol has a higher affinity for alcohol dehydrogenase and is present in higher concentrations than the antifreeze, it is prevented from binding to the active site. Due to ethanol binding there first, this counteracts the effect of the antifreeze in the body.
Potassium cyanide acts as a non-competitive inhibitor of an enzyme involved in aerobic respiration. This means that it can bind to the enzyme cytochrome C oxidase in different regions, for example the allosteric site. The binding of this inhibitor irreversibly distorts the 3D tertiary structure of the enzyme. If this enzyme is inhibited, ATP cannot be produced aerobically, since oxygen cannot be converted to water at this vital stage. This means that cells can only respire anaerobically, leading to a build-up of lactic acid in the blood. This is potentially fatal. The enzyme will become denatured if the temperature becomes too high. If the temperature is too high, chemical bonds within the enzyme will break, meaning the substrate can no longer bind to the active site. If the temperature becomes too low, the substrate will not have enough energy to move around and fewer collisions will occur. Changes in pH affect the shape and charge of an enzyme, and also affect the shape of the substrate, thus resulting in the substrate no longer having a complementary 3D shape to the active site of an enzyme. This lowers the rate of reaction significantly.